So they all are a student or
Yeah,
faculty?
they all are students. Two are BBSc and H. How many are fourth year BBSc and H? Two of them. How many are masters? How many PhDs? I forgot to count. About half and half is masters and half PhD. About nine, and amongst these are gender balance is there. 10 states are represented by the all these students. UP overrepresenting.
UP is over-representing?
Yep.
That's what you said?
Yep. We had no other certain obligations were there, but we tried to introduce the heterogeneity, like students from BHU, Aligarh Muslim University are there. Several central universities have come over here. Also, we had some obligations to take some local students, so that we have. That's the reason. One of the reasons why they're all deserving. Of course, that's not the issue. But still, when it comes to the number of students from a single state, UP is locally focused. We are well. I think we have hit the clock now. It's 2:15. Good afternoon, everybody. We are meeting at the stem cell workshop sponsored by DSTCRB, and we have already started learning about the stem cells of those cells which have two cardinal features. One is self-renewal, another is differentiation potential. And in that context, we have talked about the embryonic stem cells which are pluripotent. And today we will have an opportunity to listen in detail about another type of cells which are adult stem cells. And within these adult stem cells or tissue-specific stem cells, today's talk is on mammary stem cells. Someone who has got more than 17 years working on this particular population of stem cells, mammary stem cells. Who could be a better speaker than Dr. Ratan K.Choudhary? Dr. Ratan K.Choudhary is currently an assistant professor at the College of Animal Biotechnology, Guru Angad Dev Veterinary and Animal Science University in Punjab. Dr. Choudhary is a truly international in a sense. He has done his BBSc from Tamil Nadu and then did his master's from south to north in IBRI. He is an alumni of master of the next division, our neighboring division. And then he did his PhD from Maryland. And again, postdoc from Kentucky and University of Vermont. He's truly international in that sense. And he's currently working on two main areas. That includes one is regenerative medicine, another is veterinary diagnostics. He has published several books, also different other mobile apps, published more than 50 research articles, 10 book chapters, and presented in several forums. Currently, he is holding two externally funded projects. And ladies and gentlemen, let's welcome Dr. Ratan K.Choudhary. Ratan,
Thank you,
stage
sir.
is yours.
Thank you so much, sir. Okay, first of all, I would like to thank Dr. Sujoy sir. Um,
Yeah.
oh,
Your voice is not clear, Dr. Choudhary.
now, how is it? Is it okay now,
Low,
sir?
low, very low. Okay, if you can raise the volume.
Um, is it okay now, sir?
Better. Yeah.
Okay, thank you, sir. So, so first of all, I would like to thank Dr. Sujoy sir for giving me this platform to introduce you about the mammary stem cells. So thank you, sir. Okay, how many of you know about this peculiar type of stem cells? Um, so, so in this presentation today, we will learn what is mammary stem cells and how do we identify it, My major focus will be on bovine mammary stem cells. That was my PhD work, and I'm still working on that area. And then what is the application of those stem cells? Okay, in particular to mammary stem cells. And then I will show you some data from the lab that was generated in the DBT project, in the previous DBT projects. That was the expansion of goat mammary stem cells. Okay, and then other applications of memory stem cells, and and these works. Before I start my presentation, I would like to acknowledge some of the places, excellent places where this work was performed, or like USDA, Beltsville, Maryland, University of Maryland, from where I did my PhD, and of course my alma mater where I'm working now, and then University of Vermont for some of the work, or at least some picture that I took from those labs, and then University of Missouri where I conducted one experiment on cow. So this is a small, very small team I have, and this is the front door of my lab. Okay, so this I think you you quite aware that what is a stem cell, what is unique properties, self-renewal, multipotency, plasticity, maintenance of genomic integrity, transplantability. If I put that cell into an organ, that should able to form an embryonic body. Okay. Another property is niche dependent. Means we all have a comfortable environment. The moment, like the stem cell, if you take those stem cell out of the niche or the microenvironment, they will they will differentiate, or they will change its plasticity. Okay, so here's a comprehensive, you know, um, types of stem cells based on either it is a potential, you say, or based on the source. Okay, so this pretty much embryonic stem cells you have been studied so far, um, and so I'm working. I'm telling here to focus on the tissue specific stem cells, and so tissue specific, as the name indicates, one tissue have one type of stem cells. So if we talk about the blood, which is a tissue, then you have a hematopoietic stem cells, and then you have a progenitors, and then you have a whole lot of different cell types, and that all originates from the hematopoietic stem cells. Okay. So then you have a muscle stem cells, cardiac stem cells, neuronal stem cells, intestinal stem cells, liver stem cells, you name the organ, and there are stem cells. And finally, the memory stem cells. You have one mobile app that we developed in collaboration with the US. You are welcome to download and understand more about the stem cells. If you wanted to know how it acts, the mechanism of action, so this app is very good for you. Go to the Google app and then download it. And then if you want to know about the stem cells, in particular to veterinary science, then you have a book, you can read it. Okay. So mammary stem cells are responsible for, the growth of the mammary gland, tissue homeostasis, tissue turnover. Okay. It will replace and regenerate mammary epithelial cells. And you all know that it is the mammary epithelial cells that are responsible for secretion of milk. So, so the cow having a big udder, that doesn't mean that cow will give more milk. It is the number of mammary epithelial cells that present at the time of milking decides the amount of milk production. Likewise, also it's the secretory activity of these the epithelial cells that secretes milk. So that is, in other words, the milk production of an animal is proportional to the number of cells and also the secretory activity for cells. Okay. In addition to these roles, recently the use of stem cells in treatment of mastitis is very prominent, and it's coming up. So I will show you whatever the data we have so far in the literature. Okay, you know the cow mammary gland is called udder. So you have a left udder, right udder, and then left rear, rear, and then in front you have left front and right front. So one mammary gland is in cow is consist of four. One udder of a cow is consist of four mammary glands. Okay, and this is this the udder has been chopped off. Of course, this is we don't do in India. And if you look at the grossly, then you have these are the fat layer, which is a glistening surface. And if you cut it open, then you have it's called mammary parenchyma, okay, where the stem cells resides. Okay, if you look at the schematic diagram, then you have a gland sister, which is a sac-like structure where temporary milk is stored, and that is being squeezed at the time of milking. And then you have a different lobes and lobules, and then it is the tissue architecture is something like this, where you have lobules, and each lobules consist of a unit is called alveolus. And this alveolus again is made up of different cell types. What is most important here is the orange color cell, which is the mammary epithelial cell. So these cell secretes the milk, pour that milk into the lumen, and then there is another cell types, which is this one, grain color, that's myoepithelial cells, which is located on the basal side. So at the time of milking, this myoepithelial cell squeeze the epithelial cell so that the milk goes in the lumen, and then it comes from here, this through the duct, it comes to the gland cistern and then comes out. Okay, so you can see that the mammary gland is a very complex organ, and in that various cell types are present. If someone describes the the profile of a mammary gland by taking the tissue, basically they are profiling all different cell types. Okay, so that you should keep in mind. And this is the how the structure of a histological structure of cow and heifer mammary gland look like. You say that this is the alveolus, which is you arranged with single layer of epithelial cells. Okay, and myoepithelial cells is elongated one, something like this, is hard to, because there's very flattened cell types. Okay, and on the right side you are saying this is the mammary structure, tissue structure of a heifer, means we never give birth to a calf. So it's a different, this is you can say this is the mammary structure of a virgin calf, and this is the calf. Okay, so in this, I just want to emphasize these these small portions. So if you look at here, then it has a three different layers. The one which is here, this is the lumen, okay, which is the hollow area. And then the cell which is close to the lumen, this layer is called luminal layer. And on the outer side, this is called the basal layer. And then cell in between the luminal and basal, they are called embedded layer. Okay, so the mammary structure in heifer cow is consists of three different layers: basal, embedded, and luminal layer. Okay, and then these stroma. Okay, so these are the stroma, these are the epithelial cells, and then you have a lumen, and then this is another layer of epithelial cells. Okay, same way in the goat. Okay, so mammary structure is similar to that, and the presence of stem cell is somewhere in this epithelial region. Okay, so what I'm showing here is the presence of cytokeratin 14. Cytokeratin 14 is one of the the marker that express in the mammary stem cells, either is goat or bovine, or even in mice, or even in human. Okay. Okay. So what are the methods or ways by which we can identify the mammary stem cells? So far, I think I'm hoping that you understand the cytoarchitecture of the mammary gland and what are different cell types and mammary stem cells, which is a epithelial in nature. Okay, they are the mammary epithelial cells, stem cells. Okay, so now what are the various ways by which we can characterize or we can locate these stem cells? Okay, one is the morphological evidence. We'll know that, the stem cells are usually bigger in size. The nucleus to cytoplasmic ratio is high. So this is a cell, and if I draw something like this, and there is another cell and something like this, by looking this the morphology, I can say that this looks like a stem cell because the nucleus to cytoplasmic ratio is is high in a stem cell. Okay, so that's the morphological evidence, and that's earlier studies that came in 97 or 2002, where they say the small light cells and large light cells in mouse represents mammary stem cells, which is shown here. Okay, so at that time, that was the only concept, how, pale staining cells. Usually the cells that are not differentiated, they don't take up the stain. They remain, light in color. And those cells that are fully differentiated, they be a darker in color. So that was the earlier evidence. Okay. Then another way that initially people have identified is called side population. So increased expression, and this is based on the expression of transport membrane protein, or BCRP. Okay, so those cells, based on the site population, those cells that scattered or separated out, based on the expression of BCRP protein, they considered a side population fraction. And in human, the percentage of SP is 0.2 to 1%. And again, in mice is the same, 0.5 to 3%. And again, in bovine, it is 0.5%. So that means the population of mammary stem cells is close to, or you can say less than 1%. So that means out of 1,000, one cell could be a stem cell. So that's very a small amount, unlike that of embryonic stem cells, where you get sufficient number of cells from the embryos. Okay. Another is the multi-parameter cell sorting. So multi-parameter, the term indicates, we use different cell surface markers, and using the flow cytometry, we separate them or segregate them into different cell population. So for example, lineage negative, which is lin. So usually CD45 is a lineage marker for lymphocytes. So what we want the stem cell to not to be the lymphocytes. So, and not to be the mesenchymal in origin. Mammary stem cell is an epithelial in origin because it's a epithelial cells. And then the marker like CD24 and CD29, okay, whether it express or it express high, sometimes medium, sometimes low. So even CD24 or CD29 high, there are three different cell types: medium, high, and low. So based on the intensity of expression, it has been characterized. And that was the landmark paper that came in 2006 by Shackleton in mice, and Stingel in human. Okay, so the difference was the human. So in addition to 29, 24, CD49 came into the force. Okay, and then later in 2020, you talk about the bovine, then you have a CD49 high, which is equivalent to that, and then you have a p cadherin negative. So p cadherin or e cadherin, these are the addition molecules. So usually, this epithelial cells, because it's a lineage negative, that expressing high amount of CD49, and they are not differentiated for, you can say, for attachment. Okay, they are attachment free, so those markers should not be there. So if we take those cells out of the total, then they might represents the mammary stem cells in the bovine. Okay, CD24 is a heat stable antigen. CD29 is a beta 1 integrin, and 49F is the alpha 6 integrin. So integrin is a as a marker. Okay, and that was the paper came in Nature. So how they do, they take the tissue and then do the digestion uh, using the enzyme and make a single cell suspension and throw to the flow cytometer. Then flow cytometer has a capability to sort them out depending upon the forward scatter and side scatter, and then later based on the marker like CD24 and CD49F. So you take those cell suspension, you label them using the antibody, and then you can sort them out in four quadrant. Okay, and you can identify this is 1, 2, 3, 4. Okay, so using this, you can see that they have identified two populations completely, and this is called mammary colony forming cell population and mammary repopulation unit. So colony cell formation, this has a high expression of CD24 and CD49F, whereas this population repopulating unit MRU has a CD24 medium and CD29 high phenotype. Okay, so if you inject this cell into the mice, it will make a mammary gland. So that means this cell has the capability to repopulate the mammary gland. Okay, so that indicates the presence of a stem cell population. Okay, this is the bovine. Again, the same flow cytometry based method. You can see here side scatter and forward scatter. This is the whole population you select, and then you gate them to identify the live cells and dead cells, and then you take those live cells here, and then again you identify which are CD45 negative and which are CD5 positive. Again, you are gating it. So this is a different gating strategy. So what I'm trying to what they are going to do here is they're taking or identifying the cell. This is the total cell to have a lineage negative cell, and then in this lineage negative cell, I'm going to identify different cell types like CD49F and cadherin negative. That was the stem cell population in bovine, and then you have another cell types. Okay, so these may not be have a stem cell population, but certainly this has a high amount of stem cell activities. So now this is a lineage negative, and then you have a CD49F, which is a high because this y-axis, and then you have a p cadherin. This is negative. So if you take a graph here, then this is a E cadherin positive quadrant, and this is negative quadrant. So this is a E cadherin negative. So that's how you bifurcate the different cell types or population from the total subtypes. Okay. Okay. So how much different cell types based on the marker we identify? We identify these markers. So and then the people gave a model. Like this is the stem cell population, and then something in between. There may be intermediary cell types they do not know, and then they have a common progeny results, and then common progeny results that give rise to luminal and myoepithelial. So now you luminal, you understand that, this is the mammary epithelial cells, mammary alveoli, and in mammary alveoli, there is only single type of cells, right? And they are the luminal in nature. And the myoepithelial goes, as I said, it squeezes the mammary epithelial cells, so they are basal in nature. So in mammary stem cells, there are always two lineages. One is the luminal and other is the basal. Okay. So in the luminal progenitor, so progenitor cells are the cells progeny of stem cells. Okay. So stem cells give rise to progenitor cells, and then progenitor give rise to differentiated cells. So this progenitor luminal give rise to ductal, which is ER positive and ER negative, and then alveolar that secretes milk, which is here. So this is a one type whose origin is again from a stem cell and then from the progenitor cell, luminal progenitor cell. And then from here, you have a myoepithelial progenitor that give rise to only myoepithelial cell. So this is the bipotent and this is the unipotent, means it can give rise to two different cell types, and this one is only give rise to myoepithelial cell. Okay, so far we have studied morphological evidence based on the staining, site population, and based on the BCR3 protein, then multi-parameter cell sorting based on D3 and 4 different markers. Now comes to the in vitro study. That's called Mammosphere. So Mammosphere, the first concept came in 2003 by Don Tu. She said that if a stem cell grow in suspension culture, then they form a ball-like structure. And that ball structure, if you is made up of epithelial cells, if you dissociate those mammosphere, then each cell has a property to again produce the secondary mammosphere, something. So the concept is, if there is a stem cell, then it has a proliferation capacity, then a ball-like structure will form, and then if you dissociate them and again you regrow them, then each cell will form a ball-like structure. Okay, so that indicates this cell has a self-renewing capacity. So that means where it is originating, that is a stem cell population. Okay, and then this the real picture of a stem cell mammosphere. You can sell the clusters of cells, and this is the showing you this size. Okay, how it is done? So you take the tissue, digest it, plate it, grow in a suspension culture. Some of the cells, not all, some of the cells which are stem cell in nature will produce a mammosphere, and again you dissociate them to get the epithelial cell, single epithelial cell, and you grow them, then they will grow it. Okay, and this is the paper showing the mammosphere culture from horse, pig, cow, deer, dog, and then monkey, hamsters, mouse, rat, and human. Surprisingly, the structure of the epithelial cells derived from mammosphere was fibroblast-like structure, except the mouse. Okay, so the mouse epithelial cell structure was a cobble stone-like morphology, which is a typical feature of uh, epithelial cells. Okay, so the mammosphere was a method. Okay, so next is the label retention study. Is that clear to you, or are you getting it? Okay.
Go ahead. Go ahead. Okay. Thank you.
Okay. So next is the label retention study. Label retention study is based on the principles of asymmetric segregation of DNA, um, which I will explain a bit later in the subsequent slide. So these are label retaining epithelial cells, or LREC, was considered a putative stem cells in murine, and also our group by my advisor Kabuko, shown in bovine. Okay, and later that holds true in intestinal crypt cells, muscle cells. Okay, and in the subsequent study, we characterize those label retaining epithelial cells in bovine, using the microarray, and we showed the evidence that they are the stem cell population. Okay, so how the label retention study goes. So you inject the BRDU, which is a bromodeoxyuridine, which is a nucleotide analog during the replication or DNA synthesis. These BRDU get into the DNA. Okay, so you inject the animal, and those stem cells, if they are dividing, they go into the cell. So inject the BRDU. So in the system, it goes, and during the replication, you get the BRDU level DNA. Okay. And when is cytokinesis, and then you get both the cells have a BRDU in the nucleus. In the subsequent mitosis, then you don't give the BRDU. So let them allow to divide and divide and divide and let get rid of it. That's the dilution of the BRDU label. Okay, so now new DNA synthesis, there is no BRDU, which is indicated by a yellow bar, no BRDU, but it's still, since BRDU is present in your cell, so that will be there. So if there is a random segregation, then it will divide into two daughter cell, and the label will be bit later because BRDU is getting diluted. But if there is a non-random segregation of this cell, then the BRDU remain with the one daughter cell, and the other daughter cell will not receive the DNA. So based on that, we identified the population of label returning epithelial cells from the basal location and from the epithelial location, which is diagrammatically shown here. And then we dissected those cells out using the laser macro dissection microscope. So and we collected those cells in the different tubes, and then we subjected to microarray analysis. So the gene expression analysis shown that the HNF4A, which is a liver stem cell marker, what at the hub, and those stem cell marker genes were upregulated in these label retaining epithelial cells that was located in the basal region. And finally, we showed that based on the expression of those stem cell marker gene and its locale in the basal, we hypothesized that they are the stem cell rich populations. Whereas if they are more apically or located or in the embedded layer, then the stiffness goes down, and they are considered as the progenitor cells in comparison to non-living cells. Okay, and then at the end we provided that the stem cells are liver retaining cells are located in the basal layer, and these are the different new markers we proposed in bovine. And later, that was in 2009 paper, you can see it was very evident in mice also. That's holds true. So now it's instead of BRDU, now the safe marker is EDU. You can label them EDU, and they chase from 2 week, 8 week, 10 week, and 11 week. You see still that is there. The EDU label cells are there in the mice mammary gland. Okay, so these are the new markers we suggested: NR5A2, NUP153, FNDC3B, HNF4A, which is very pertinent to the literature that shows they are related to the pluripotent transcription factions and stem cell activity. Later, based on the macro data, we also confirm whether those genes are expressing at the protein level. Very well, happy to share that NR5A2, you can see very few cells that are intense in color, that is labeled using these 4 markers in the prepubertal mammary gland. Okay, and then that was consistent in the cow also. So this is a one mammary alveolus. You can see presence of a single stem cells or progenitor cells make here is worth to investigate. Further, we quantify the percentage in cow and calf, and you can see there's a very big difference. Is good to know here that this is a marker of stem cells as well as the progenitor cells. So here at peak lactating cow, the number is more, indicating, suggesting that, it is the progenitor cells, which is a trans-amplifying cells, that might be more in the lactating cow, because the cow is in high demand to produce the milk. Okay, and again also we see their expression from peak lactation to late lactation to mid lactation. It's a decreasing, so which is consistent. So therefore, the the dry period is given to the cow. It allow the cells to regenerate, the stem cells to regenerate and proliferate to have the milk production in subsequent lactation better. Okay, and this is the co-localization study where whether the LRC, the label returning cells does express our marker. So is very, you can see that they co-localize each other. So this strongly give an indication that the stem cells, the label retaining cells, also express these novel markers of bovine mammary stem cells. Okay. And this is the expression of classical markers, which is ALDH1, aldehyde dehydrogenase 1, not very good marker in bovine. However, it's very good marker for cancer, mammary cancer, and all those. Another marker is MUSASI1, which is quite good. Okay. So after this label reading experiment, a new experiment, which is called linear tracing experiment. So where you can label the different cell types with the different colors, and you can chase them after the differentiation or lineage commitment, where the cells are, what they are making it. So that's the another model to study the stem cell population, especially in the mouse. So using this lineage tracing experiment, they have found LGR5 and tetraspanin 8. They are the quiescent subsets of cell and represents the stem cell population in mice. But that may not hold true in human. In a human, they found P63, cytokeratin 14 and 19 positive cells. They are quiescent putative stem cells. Okay, so once this LGR5 expression is go down, then that becomes progenitor cells. Okay, and if the another marker it express proc R, then that quiescent stem cell becomes cycling stem cells. Okay, so now so see the hierarchy of stem cells. It's whether it's a quiescent or cycling or progenitor, bipotent progenitor, unipotent progenitor, and many more, which we yet to identify. Okay, so here is the another model, which is came in 2020. So model A say that during the embryo, we have the mammary stem cells or fetal mammary stem cells, and then when it comes to the adult stage, means postnatal stage, they become quiescent, and then they give rise to basal progenitor and luminal progenitor, and then various cell types of the mammary gland. Where model B says that there is no quiescent stem cells, and during the embryo, the lineage restriction occur. So that means two different cell types, luminal or basal, happen during the embryonic stage, and in the adult stage, they comes to a progenitor and basal progenitor and give rise to different cell types. Okay, so maybe both holds true, and we do not know. Okay, so in addition to that, as I said, it's not only the stem cells are not static. So when stem cells, what we are characterizing now, it doesn't hold, have the same amount of or same number of marker expressing on the cell surface. They're very dynamic. So there is a transition from quiescent to multipotent based on some stimulus like presence of steroid hormone, and then from multipotent to multipotent progenitor, which are basically a negative to estrogen receptor. So this is the hormone non-responsive cell. So hormone non-responsive cell, which further give rise to hormonal responsive cell, which is a luminal, and hormonal non-responsive cell, which is a basal. So it can go to luminal and basal depending upon the expression of estrogen receptor. Okay, so this is the, so far what we understood is this is the basement membrane. Mammary stem cells reside in the basal layer. Progenitor cells are more apically located, and these are the cells which secrete smell. Okay. So, but the thing is, we do not know whether the stem cells is in direct contact with the basement membrane, because all the cell signaling molecules or signaling things or pathways goes through the basement membrane, or the cell make a protrusion and it doesn't allow the stem cells to touch the basement membrane. Okay, so this we do not know yet. Okay, so the stem cells can differentiate into two ways. One is if it divide perpendicular to the basement membrane, which is red bar, and blue bar is the basement membrane, then you see the ductal growth occurs. So it's two cells in a form in a parallel to the duct. So ductal growth occur. But if the plane of division is in parallel, then the progenitor cell happen. So that means stem cell orientation of the symmetric and asymmetric division of stem cell depends on the cues, whether the ductal growth has to happen or whether it has to replenish the mammary epithelial cells. So during the milk secretion, when there is a worn on turn of the cell needs to be replenished. At that time, the cell divide in this fashion, where the stem cell give rise to progenitor, and then progenitor give rise to differentiated mammary epithelial cells. Okay, but if the expansion has to occur, elongation of the duct has to occur, then it divides in a perpendicular fashion. Okay, apart from that, there is also the reprogramming of cell goes on, and a beautiful paper that's showing here is the if the notch1 involved, then the basal becomes luminal, and if P63 is involved, then the luminal cells reprogrammed into basal cells. So now there's a big confusion. The confusion is that the moment we are saying, so that means the luminal, so if I go back, so that means the luminal can also come forward and divide and give rise to basal. So it's a both the direction, so all depending upon what kind of the signals the cellars getting. Okay, and the last approach to identify mammary stem cells are the single cell RNA sequencing. Okay, so based on the expression of c-Kit, c-Kit is a proto-oncogenes. c-Kit positive cells are basal, which is also keratin 14 and 18 positive. And if it's negative, then they are as the luminal cells. Based on the expression of these markers, it can be classified into basal, mature luminal, and alveolar precursor cells. This is a pretty a new paper which came into existence. Okay. So, and this is the last and latest model of showing the hierarchy of the mammary stem cells. Okay. So this model says before the birth, there's a fetal mammary stem cells which are c-Kit positive, and then after birth, there is as a pool of cells. They are trans-amplifying cells, and this pool contain the alveolar, or you can say the luminal part, and then there's a basal part. In the basal part, there are two different cell types. One is induced mammary stem cells, which can give rise to myoepithelial cells, or also it can give rise to primed or primed or called facultative mammary stem cells. This facultative can give rise to alveolar. This alveolar can give rise to ductal. This ductal can give rise to another alveolar, which is ER positive and negative, and then so on. They can change the intermediate to alveolar, alveolar to intermediate. So this compartment is very dynamic, and then finally it gives rise to ductal or myoepithelial cells. Okay, so this is the latest model we can have. So it's a very dynamic. So identifying a stem cell population uh, using giving a marker say doesn't say anything. It's very dynamic depending upon the stage of the lactation, age of the animal, and so on. Okay. So after covering the stem cells, how it is identified using various methods, I would like to focus on what is its application. Okay, so one application is certainly is the increasing productivity, milk productivity. Okay, which is shown here. Okay, another possible use can be dry cow therapy. If we inject those stem cells in the dry period, we can increase the stem cell population so that we can increase the cell turnover, or we can repopulate more number of epithelial cells for more milk production in the next lactation. Apart from that, it can go to mastitis, treatment of mastitis, because we all know stem cells has immunomodulatory properties. It has a, it functions also via the parakine fashion. It can have a antibacterial, antiviral. Um, it's also antifibrolytic effects in addition to angiogenic effect, by promoting growth of the cell and many more. Okay, and then it's also promoting the wound healing. So that's the possible applications. And okay, if you read one to more, you can go through this paper that talk about how the mammary stem cells can be used for production purposes. Okay, and there is another paper which is that is the how the health of the uh, mammary gland be uh, can be improved using the mammary stem cells. Okay. Um, so in all these, as I explained earlier, these number of stem cells are very low, 0.1% or 1%. So the expansion is required. So either we do the in vitro expansion or in vivo expansion. So this paper talks about the mammary stem cell expansion for animal production. Okay. So during the dry period, the number of epithelial cells goes down by 50%, which is a huge. So therefore, injecting mammary stem cells and increasing its population of those milk producing cells could be considered as a dry cow therapy. Okay. Um, so how that will work? So imagine that you have a stem cell population. If you increase the more number of stem cell within the gland, then you are also amplifying the progenitor cell populations, which are the trans amplifying cells. Okay, so more stem cells, more progenitor cells, and more progenitor cells will give rise to more differentiated cells, and then more differentiated cells certainly will give rise to more milk. So that's the hypothesis. Okay. 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 Um, so based on that, we conducted an experiment was a DBT project where we attempted to increase the milk production in goat using genthasin, and still we are working on that. So in this, what we did, we conducted two experiment um, in the goat in the early lactation. on 5th day and 7th day, we injected xanthocin, and then we recorded the milk production, and then followed dry period, and then again we recorded the milk production in the next lactation after kidding. In the second experiment, what we did, we injected the xanthocin for 3 days, morning and evening, and then on 10th day, after 3 days of the last injection, we took the biopsy. Milk recording is went as per the earlier experiment, and then we study the gene expression, protein expression of injected mammary cells, mammary tissue, and compared with the control gland. Okay, in that we found, we were able to localize the stem cell population. We quantify them. We also quantify the fold increase in different cell types, like this is the casein, beta casein, showing the expression estrogen receptor alpha, and how its expression goes up in comparison to the control gland. This is the differentiation marker, mutation 1. This is the LALBA, alpha lactoglobulin, and this is again stem cell marker, ALDH1 and P53. So, so the xanthocin was able to increase the transcripts of some of the markers, which is relevant to the milk production. Okay, and not only that, we also quantify CD10, equidarin, MUC1. These are all the different cell marker, and quantified cytokeratin, 19, vimentin, and you see the expression was different in control, and different all these treated animals. Apart from that, we also quantified other markers at the protein level, and then we also measure the expression on the using real-time PCR. Very well, you can see the expression of NR5A2, which was a novel mammary stem cells marker in bovine, was up. LDH1 again up. Casein was beta casein was up. LALBA was up. So these are the favorable thing. We want to have a high expression of milk protein and milk associated or fat synthesis gene, uh, and a stem cells marker. So xanthocin is really helping for cell differentiation or increasing the stem cell population, uh, at least at the gene level. And then that's the paper. If you want to read on those, you can go. These are papers that was published based on that. So finally, um, not very prominent, but we were able to increase the milk production at the rate of 2% per gland per animal. So that means if you consider two gland per goat, then 4% per animal. Okay, so still few shortcomings of this project was I took the wrong time. So early lactation is not a right time, because the system of the milk production is so optimal that no matter what you do, you're not going to increase the cell number, because cell number is already increasing by default in the system because the milk is going up. I should target the whether when there is a decrease in epithelial cell population somewhere in the late lactation or maybe during the dry period, that would be then I might have a very good response instead of two. I might have a better figure. Uh, and then another interesting thing we found in this was all the treated gland even after a surgery resumes the lactation in comparison to control gland. So only 4 animal was able to recover because of the fibrosis that caused by the mammary biopsy. Okay, so in other words, so xanthocin was helping for restoration of the goat mammary gland, increasing stem cell population, and also the functional differentiation of the mammary epithelial cells, and therefore the the milking was restored even after the biopsy. That could not happen in the control gland. So that we are assuming. Okay, so another example applications of mammary stem cells I would take from the literature, and that was on chronic on acute mastitis. Okay, so if you want to read more about the role of stem cells in mastitis, there are a few papers that I would like to suggest. You can go. So so this paper, what is saying? First, they are injecting the Staphylococcus aureus and trying to establish the mammary infection. Okay, and after establishing mammary infection, they are injecting the stem cell population. Okay, then inject 2.7, 2.7 million cells per quarter. Um, and very well, you can see that the colony formation unit of the bacteria was down. So the gray one is the stem cell injected quarter, and this red one is the control quarter. So especially in this, from day 6 to day 10, the number of bacteria was significantly reduced in the stem cell injected quarter than the control quarter. Okay, and this brown, not brown, it says orange one is the antibiotic injected quarter. So that was in between the positive control and negative control. The response of the stem cell injected, which is 25 million cells, was in between the two. So that means, it also depends the number of dose, amount of stem cells present in that dose, and also the frequency of injection. So all those are needed to have a very good positive response. Okay, so this study quite promising, that showing the utility of mammary stem cells for mastitis. And mastitis is a huge burden to our dairy industry across the globe, not only in India. You're surprised to know the incidence of mastitis is 30 to 40%. It's not 5 or 10%, 30 to 40%. And then the loss incurred by mastitis is huge. And rampant uses of antibiotics is giving rise to antibiotic resistance issues, AMR. Okay, so this could be a boon if for for the scientist, if some uh, medicine out of this system cell could be derived for the mastitis purpose. Um, and this is on the goat, and the treatment of goat with the chronic mastitis. The chronic mastitis indicates that it's not only the infection is present, but also the memory, the tissue is fibrosed and hardened, and all the epithelial cells lost secretory activity. And then this paper describes these adipose tissue-derived stem cells restore the milk production. So that means it has a fibrolytic effect. Okay, and and this is the methodology how they did on the goat. And this, you know, and each gland, say goat has the 2 glands, cow has a 4 glands, and they are anatomically or physiologically separated by a, a barrier. But surprisingly, the stem cell was able to cross the barrier. So this is the showing you, this is the injected quarter, and this is the control. No, this is the control quarter. This is the injected quarter. So this adjacent quarter, you can see the migration of those green label cells using QDATs was also there in the control. Okay, so considering this, if I come into my my experiment using genthicin, so it was possible that in my case also genthicin treated gland and control gland was the same from the same animal. So injecting genthicin using this might have, they infuse into the mammary gland, and therefore the response might not be as evident as I could have. So that's another thing we should do next time. We should keep those treatment animal completely separate from the control animal. Okay. Um, okay. So, and the possible reason how this is happening here, because the injected stem cells also secrete growth factors that has immunomodulatory functions, and that they may stimulate the resident cells to progress for the cell cycle, or maybe for the uh, growth of the epithelial cells. Okay, so with that, I think I covered applications of mammary stem cells, especially in mastitis and also in increasing milk production. But remember, they are only at the experimental stage. So none of them are in the practice. Um, and these are the group that are working on this mammary stem cells. Australian USA group, Australian group, Viswadhar, and Paul Bhupinder Paul, they're working, but basically they're working on mouse. If you come to this Italian group, they are working on mammary cancer. This is another US Max Vicha. They are working on the again mammary cancer, human side. This Canada group is Stingel, remember 2006 paper that came. This is the guy, the Canadian group, and also from the China group working on mouse, and the one paper I shown, the PROC-R positive marker expression of PROC-R gives the cyclicity of the stems, quiescent mammary stem cells. So this is the group, and this is the bovine group, very poor group. Um, uh, Me and my advisor are working in those areas. Okay, so this is the few keywords. If you look to the literature, mammary stem cells, then, responsible for gland development, mammary gland, steroid hormone, lactation, plasticity, cancer stem cells, because mammary stem cells can give rise to mammary cancer stem cells. Metastasis, mammary development, self-renewal, and of course the bovine. So what I'm trying to show you is the role of mammary stem cells in bovine mammary development for lactation somewhere is possible. So that's the point I want to make it here. Thank you so much. Now I'm happy to
Thank
answer.
you. It's wonderful.
Thank you so
Dr.
much.
Choudhary, it's a very historic as well as the whole journey starting from how we treat the one kind of adult stem cells and its entire journey we have been experiencing. I have been experiencing at least. Thank you very much for bringing the entire whole scenario into a single presentation. That's a fantastic. That's wonderful. If we have any questions, somebody,
Ask questions, ask.
yeah.
Yeah.
Good afternoon, sir.
Yeah.
My question is, if this mammary stem cells can be transformed into cancer stem cells, means then why we have to use this in therapeutic applications? Because as per your statement, this mammary stem cells having the angiogenetic property and migratory property, and it is having the high expression of BRCP gene. So if there is any mutation in this gene, means it will be prone to this cancer formation. So these and the origin also epithelial origin, it may undergo epithelial mesenchymal transition. So all these factors suggesting it may form the cancer stem cells. So if we are using this in therapeutic applications, means this is
Contradictory to why we should use for therapeutic
And
applications.
is there any marker difference between the cancer stem cells isolated from mammary tumor and mammary stem cells? And till what passage we can culture these mammary stem cells?
Okay.
Too many questions. Okay.
Yes. So the first question, if stem cell is a RAM, then cancer stem cell is a raw one. So both have the same capability. Only aberrations in cell cycle and those control works. Okay. And one of the good property of a mammary stem cell versus cancer cell, and if you see the pluripotency, they have very equal pluripotency. There's no difference in that, except that control part. So the control part is there with the stem cells, no with the cancer stem cells. Okay. And And all the adult stem cell, the property is they by default they divide asymmetrically. So whatever it's required, it takes that and it doesn't take it. But if you divide cancer stem cell, they won't divide asymmetrically. That control is lost. So they divide into two cancer stems. Okay, that's one thing. Second thing is not it doesn't work that way. There is a beautiful experiment. If you inject normal cell into cancer's tissue, the normal cell will become cancer. In vice versa, if you take the cancer cell, inject into normal cell, chances are there because of the epigenetic regulation that becomes the normal cell. Okay, so if you're taking the normal stem cell injecting into the normal gland, uh, I doubt that will create a cancer. Okay, that's pretty safe, and people have proven it number of experiment, and so they are safe. And what other questions you have? You got my answer?
Yes, sir.
Okay.
The marker difference between the cancer's stem cell isolated from mammary tumor and mammary stem cells. Is there any
Yeah,
difference?
you have to go for say FNDC3B is one of the cancer marker. Okay, so if that expression is very high, number one, and also if the expression is aberrant, say instead of cytoplasmic, if the expression goes to the nucleus, then that is aberrant expression. So high expression and aberrant expression of the same marker, that you can identify that this is a normal or cancer. Okay.
Okay. I think we could invite for a cup of tea for you, but it's
I think I finished on time, I guess.
maybe one quick question. So you already crossed the time, but we'll still have we'll take one question from Shweta.
Yes.
She's from Dr. Manoj Kumar Singh's lab, NDRI.
Okay.
Huh? Oh, Naresh is okay. Sorry.
Good afternoon, sir. Thank you for the information. Sir, I have few questions related to the
Restrict
application.
only one or two, okay?
Well,
Because we have to go to the other building
Okay,
actually.
sir. So, like, what are the different methods which we can use to introduce the stem cells to the target area? And the second question is that if we are introducing the stem cells at the area, so is it effective till lifetime or we have to give it like a doses? And if once we have given, is there any chances that the future infections will be there? And if yes, so how much more or less than a normal infection without stem cell therapy?
okay, your question is limited to the route of administration. Okay. You can give intravenous. Okay, so your stem cell preparation should be sterile. It should not have any infection that you have to make sure. Number one. Number two, if you can inject intravenous, you can inject topical, you can inject intramuscle or intra parenchymatous, intramammary, or even subcut, depending on what kind of response you want. So that needs to be tested. If you inject intravenous, so the stem cell has a homing mechanism. So it will go to the organ where it is required, or especially is go to the tissue from where it has been derived from. Okay. And of course, the dose will lose because to reaching to the distant organ through the blood, cell will lose here and there. So that's not a very good method, but still people are using it. Okay. In my case, I use the intramammary injections. Okay. So the question is whether all the stem cells will reside and survive and repopulate, that we do not know. That's the one mechanism that how it works, but there is also other mechanism like paracrine factors. Okay. So they reside or they talk to the neighboring cells, the paracrine factors. So that's also infused. Some cells may fuse with the resident cells. So that's another mechanism. You go to the app and then your question will be solved. Okay. And regarding infection, how many doses, that upon the severity of infections. If you have a very, say, very acute and severe, then you have to repeat 3 and 4 times. If you have a mild, then 1 dose should be sufficient, maximum 2 doses. Okay. For 1 dose, in the US they are charging $1,000, 1 million cells. So 25 million cells. Imagine how much people are making money to. Okay, thank you.
Okay, we'll close here. Thank you very much, Dr. Choudhary. And I will encourage I will share the app of yours to
Yes,
the
sir,
group
that would be good.
for those people, those
Sure,
who
sir.
have not yet seen it. It's a very informative. I have seen. I haven't look at it. Wonderful. Again, we thank you on behalf of everybody and also